So karyotype describe, the title define a karyotype. It describes all the chromosomes in the nucleus. We have 23 chromosomes, 23 pair of chromosomes. And of the 23 pair of chromosomes, 22 pair we call it autosome. Autosome because they are specific to the body. And the 23rd pair, the last pair, we call it sex chromosome, the X and Y. X and Y denotes male, and then X and X are female. And then we call them homologous pair or homologous chromosome because one chromosome, they are from the father, paternal. And then another one is the maternal, from the mother. Therefore, we call it, they are one pair of homologous chromosome. And in other words, since you have 23 pair of homologous chromosome, then we will have 46 chromosomes. So remember, in the exam, if I ask you about pairs of chromosome or number of chromosomes, we have 46 chromosomes, but we have 23 pair of chromosomes. Now let's start with mitosis. Mitosis, as we learned from the cell cycle before, most of the time the cell, they are in the interphase. And then starting from prophase, and during the interphase, specifically the S phase. S phase is when the DNA replicates. Now after G2 phase, then the cells start to go into mitosis. We have prophase, metaphase, anaphase, and telophase. So the main purpose of the mitosis is to separate the sister chromatids so that you have one chromosome for each daughter cell. So that's the main purpose, main idea. And in Bio 3B lab, you will have a chance to look at the animal cell and also plant cell. Animal cell from a fish, I believe, and then the plant cell from onion root tip to identify different phases. And it is something important for you to know how to identify each phase. So in the interphase, what is the characteristics of interphase? If you look under the microscope, interphase has the cell in the interphase, they look regular cell, meaning that you will see a black dot with a very concrete boundary surrounding it. This black dot is the nucleus. So that's it. This is the interphase. What happens is that during interphase, inside the cell, you still have chromatin inside the cell, and you have the nuclear membrane, nuclear envelope surrounding the nucleus. That's why the boundary of the nucleus is very sharp, and it is very dark because they are all chromatin filled in. And then in the prophase, what happened in the prophase? They divide prophase into early and late prophase, but normally if you look under the microscope, you cannot tell the differences between early or late. In general, in the prophase, you will start to see chromosomes. But under the microscope, what you see is that it's a black dot, but in the black dot, you have some white empty spaces in the black dot. That is because the white empty spaces, because all the chromatin, they coil up and become chromosome. When they become chromosome, then you will see a lot of empty spaces inside the nucleus. And another characteristic is that the nuclear envelope dissolve or disappear. So you will see, you can still see a kind of like a nucleus with a lot of white spaces, but the boundary of the nucleus is fuzzy. It's not very sharp and clear, unlike the one in the interphase. In here it says that the centriole, the centriole, it tells you that the spindle from the centriole the central start to migrate to the opposite side of the cell, but in the microscope, you won't be able to see that. And theoretically, the spindle from the central, the spindle, they are the cytoskeleton. They are the they are the not actin, not intermediate, they are the microtubule. They're the microtubule from the cytoskeleton. So they're the largest in size of the cytoskeleton. So there are three types of cytoskeleton, just for review: actin, intermediate, and microtubule. So this is microtubule, this spindle is microtubule. In the prophase, the microtubule attached to the centromere of the chromosome. As a reminder, centromere, they are the ball-like structure in the chromosome. Therefore, attachment of the microtubule spindle. But again, the spindle attached to a centromere is something that you cannot see from the microscope. So it tells you that, okay, it is microtubule, centromere, microtubules, they're the spindle, and they attach to the opposite side of the centromere. They said that specifically to the protein called kinetochores of the centromere. That is okay if you want to know that. So what happened is that right now they are undergoing something called a tug war. It's like a tug war, meaning that the spindle, the microtubule, they are pulling from each side, either elongating or shortening from the opposite end. So that it's like a tug war. They're juggling right now. When all the chromosomes, they align in the center of the cell, we call it metaphase. And in the bio3D lab, when you look under the microscope, metaphase should be the easiest mitotic phase that you can identify, because you will see a black line in the middle, just like this in here. And then after metaphase, what happens is that the spindle or the microtubules shorten to the opposite end. When they shorten to the opposite end, it literally pulls the two sister chromatids apart. When they pull apart, when you look under the microscope, it looks like this. It actually looks like the two hands, they're facing each other, the fingers, they're facing each other like this. When you see it like this, when they separate like this, then it is the anaphase. Telophase, it really depends on if it is an animal cell or plant cell. If it is an animal cell, telophase is characterized by this. We call this a cleavage furrow. Cleavage furrow is basically the invagination of the cell membrane, starting of the invagination of the pinching or invagination of the cell membrane, and you can see two separate nuclei on the opposite end. So it is like two cells, but it is still one cell. It's like two cells connected to each other in the form of one cell. Then we call this a telophase. And they said cytokinesis continues, and then you will go to G1 phase. So cytokinesis is a process of dividing parent cell cytoplasm into two daughter cells. So that is the definition. What happened in the cytokinesis? What causes the invagination or pinching of the cell membrane to form the cleavage furrow is that the actin filament from the cell, they start to contract, and then they pull the plasma membrane together. And that's how the cleavage furrow is formed. And cytokinesis is the last step in the cell cycle.